Hi guys, welcome to Cosmic Solutions, Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. So in our previous session on cell cycle and cell division, we had discussed about mitosis, what are the different events that occur in mitosis, what are the different phases, what are the significances or what is the significance of mitosis. So all these things we had seen. So in today's session, we will see what is meiosis. Okay, and how it is different from mitosis. What makes it so special that we had to create a separate category or separate um, topic to study this process. Okay, so let's get started with meiosis. So basically, what is this meiosis? Okay, so meiosis, okay, its main thing or the one purpose why meiosis occurs in human body is production of gametes. That is the sex cells, the sperm and the egg. Now we know that the 50%, there is 50% contribution of each parent in formation of a individual. So when we are talking in terms of chromosomes, we know that exactly half the chromosomes would come from a father, half the chromosome would come from a mother. So this forms the basis of your XY sex determination. Okay, so we know that an XX set of chromosome will form a female, whereas XY set of chromosomes is going to give rise to a male child. So this is where your meiosis comes into picture. Okay. Now meiosis in human, okay, it's a division process, okay, to form haploid cells. Okay. Your process will start with a diploid cell giving rise to haploid cells. Okay. Now if we go back to your mitosis, we know, okay, cell division, it is made up of two parts. There is an interphase and the M phase. So M phase is actual mitosis. But before entering the mitosis phase, a cell has to undergo the interphase. So I gave you an example for bus stop. Interphase is nothing but like a bus stop. You are actually preparing for your journey. Whereas your mitosis is the actual journey. Same thing happens in case of your meiosis. Okay. So before entering the meiosis, a cell has to go through the interphase where there will be cell growth, okay, during G1. Then there will be copying or the replication of your genetic material or the DNA during the S phase. And the final stage where you are actually ready to pick up your luggage and enter in or board your bus, that is your G2 phase. That is the actual preparation for your division. So a cell has to undergo all this process of your interphase before entering the meiosis. Okay. So now let's see what happens in meiosis. So if you recall back, okay, in case of your mitosis, if the parental cell had 2n number of cells, okay, the progeny cell or the daughter cell still would have the same number of cells. That is, if the mother had 2 and mother cell had a 2 and number of cells, the daughter cell, okay, or the progeny cell formed, it is still going to have the same number of chromosomes. So this made it an equational division. But in case of your meiosis, okay, it is called as an reduction division because, okay, there is reduction in the number of chromosomes by exactly half. That means if a parent cell had 2n number of chromosomes, okay, the daughter cell will have n number of chromosomes. So that means it is getting reduced by exactly half. Okay. Then here there is, in this case of your meiosis, it makes sure that haploidy is formed, or if you say this is diploid, that is 2n, it gives rise to a haploid phase. Okay, and this is very important for sexually reproducing organisms. Okay, now if there is fertilization occurring, between, suppose now we have a cell having this N, and suppose we have another cell, okay, having another set of N. That means, suppose, let's see, this is the egg cell from mother and this is sperm cell from the father. The union of two, if there is a fertilization, it will give rise to a genetic makeup, okay, which will have the chromosomes from both the parents. That is, it will have 2N. So that is a cell, it will start with a diploid cell, it will give a haploid phase, and if there is fertilization, it is going to give rise to a diploid zygote. So which will be contributed equally by both the parents. So same way, when we are talking about sex determination, one sex chromosome, okay, would come from your father and another would come from your mother. So X will come from mother, Y will come from a father, giving rise to a male child. Whereas if both the parents are contributing for XX chromosome, okay, it will give rise to a girl child. So this is the beauty of meiosis. So why there is requirement of this haploid phase? The reason is this, because this is occurring into the cells which are essential for reproduction. Okay. And then this meiosis occurs during gametogenesis in both plants and animals. Okay. So now let's see what are the key features of meiosis. What makes this meiosis special? Okay. Now important points here. Okay. There are two sequential cycles of nuclear and cell division that occur in your meiosis. So we can classify them as meiosis I and meiosis II. Now, important point from your NEET as well as your exam point of view, board exam point of view, that is, there is only one cycle of DNA replication. Though there are two sequential cycles of nuclear and your cell division, okay, but there is only one cycle of your DNA replication. Then meiosis I is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. So this is a prerequisite for meiosis I, that is, okay, production of parental chromosomes with identical sister chromatids. Okay. Now, the meiosis, okay, there is one thing that occurs, that is pairing of your homologous chromosomes. And then there is one new term that will come into picture is recombination. So, we'll see what is recombination in further slides. Okay. And here totally different thing that has happened over here as compared to your mitosis is that there is formation of 4 haploid cells instead of 2 diploid cells which had formed at the end of mitosis. So these are the key features which differentiates mitosis and meiosis completely. Fine. So now let us see some more things. Now before we move on to actual meiosis, the processes of meiosis, okay, let's see what happens, okay, or let's get familiarized with some of the terms, that is your haploid and diploid, okay. So in the previous session also I had spoken about something called as haploid and diploid. Then I spoke about homologous chromosomes, okay. So when we were discussing about mitosis, we have seen what are sister chromatids, right? So now these are the new terms that have come into picture, that is haploid and diploid. So let's see what is meant by haploid, diploid, so that when we go ahead, okay, there is not much confusion, okay, because usually these things they become very confusing. Now when we talk about a haploid cell, okay, whenever we are talking about a haploid cell, if you see this picture, okay, Now this is a haploid cell. You see it is denoted by N, whereas the diploid cell it is denoted by 2N. Now in the picture, the cell picture, if you see, there is only one set of chromosome, whereas in case of diploid there are pair, there is a pair. Okay, there are two sets of chromosomes. So a haploid cell contains only one set of chromosome, whereas diploid there are two sets of chromosomes. So these haploid cells are the result of your meiosis. Okay. Then next is homologous and non-homologous chromosomes. If you see this picture, okay, from this picture itself we can make out the differences. Homologous, if you see lengthwise also they are same. The centromeric position is also same. Non-homologous, if you see lengthwise there is a difference. Okay, in some cases there may be difference in the centromeric position also. Okay, so we'll speak about some more differences also. Okay, now in case of your homologous chromosome, it's a pair of chromosomes with same sequence of gene. Whereas in case of your non-homologous chromosomes, they do not form a pair. Okay, they do not constitute the same pair. Okay, then structure wise, both arms of chromosome centromeric positions, they are the same. In case of your non-homologous chromosomes, there may be differences in this. Then when we speak about the alleles of the same gene are present at the same locus in your homologous chromosomes. But in case of your non-homologous chromosomes, these alleles they are of different genes. Okay. Then why they are important? Because okay, your chromosomes, okay, no, your homologous chromosomes, okay, they belong to the same pair of chromosome, okay, comprising of parental and your paternal and maternal chromosomes. Whereas in case of your non-homologous chromosomes, the chromosomes they belong to different homologous pairs. Okay. Then your outcome, when we talk about the outcome, okay, in your homologous chromosomes, okay, this is essential for your recombination. We'll see what is that. So here, some part of chromosomes, they could get exchanged. Whereas in case of your non-homologous chromosome, okay, it comes into picture in case of your translocation. Okay. So if you see example, okay, all the 22 autosomes, okay, the non-gametic cells, okay, they form the homologous chromosomes. Whereas your example of non-homologous chromosomes is your X and Y chromosomes. They are totally different. So that forms your non-homologous chromosomes. So I hope with this most of the things about your diploid and haploid and homologous and non-homologous chromosomes is clear. So I expect that in future there would be no confusion whenever you will come across these terms. Because as a student, I always had confusion about these, especially homologous and non-homologous chromosomes. Okay. So now let's move on to the next slide. So if you see this slide, okay, here we can see that meiosis I and meiosis II. So picture wise, if you see, we do not see there is much difference. Most of the things they are the same. Okay. So there is an interphase at the start of your meiosis I. Okay. So if you see this picture, okay, a diploid cell is entering meiosis, that is bearing 2n number of chromosomes. So same just like in case of your mitosis, there are phases: prophase, metaphase, anaphase, telophase and cytokinesis, but there is a number 1 attached after each term. So that is an indicative that it is a part of meiosis I. Similar processes are occurring, okay, but there are some different things which we'll talk about later. Then next is your meiosis II. Again, if you see, the terms are similar but with this number 2 attached. So that is, okay, it is a part of your meiosis II. Okay, so as we go ahead, we will discuss more about it. Now here I want to bring it to your notice that the journey of this diploid cell, that is 2N, it has become N or the haploid. Okay, so meiosis I, you from a diploid cell, you get a haploid cell, two haploid cells, and these two haploid cells, they will enter the meiosis II, okay, giving rise to four haploid cells. Okay, so now let's get started with the next phase, that is the meiosis I. So meiosis I, the purpose or the role of meiosis I is to separate the pair of homologous chromosomes. Okay, so it has to separate the pair of homologous chromosomes and reduce the diploid number to haploid. So diploid to haploid transition occurs in case of your meiosis I. Now meiosis I is further subdivided into four more categories just like your mitosis. Okay, that is your prophase I, metaphase I, anaphase I and telophase I. So now let us see what happens in your prophase I. Okay, now this prophase I, okay, it is longer and more complex, okay, as compared to the mitosis of the prophase of your mitosis. Now because it is longer and complex, okay, it is further subdivided into five more subcategories, okay, based on the behavior of the chromosomes. So we have leptotene, zygotene, pachytene, diplotene and dikinesis. So these are the five subdivisions or the subcategories of your prophase I. So let's see what is there in each one. So now to remember this, now it is very important that you recall or remember these things in a sequence because each phase is correlated to the next phase. So if you jumbled up with the phases, it is of no use. So how do we remember it? So I have made a simple way, okay, to remember this. Okay, this is a totally original thing. Okay, I'm sharing with all of you. That is, we have to, I remember it as lazy panda diet. So I have nothing against animals. Okay, just because I'm associating panda diet. So do not blame me for this. Okay, this is just to remember. Okay, L stands for your leptotene. Okay, Z stands for your zygotene. Okay, ZY. Then P stands for your pachytene, PA. Then DA, okay, it stands for your diplotene and DI, okay, further it stands for dikinesis. Then again you will say here also there is DI and here also there is DI. Okay, so how do we remember it? Okay, so if you see here diplotene, DIP, and this is pachytene. So this pachytene and dip will come one behind the another. So that is P is here and there is P over here also. So it is sequence can be easily remembered. That is leptotene, zygotene, pachytene, diplotene, dikinesis. So if you remember this lazy panda diet, I guess you will remember this forever. So there will be no confusion as far as the sequence of this subdivisions is concerned. Okay, so let's move on to the next part, that is okay how this transition occurs from your meiosis, okay, in your meiosis. Okay, so we were talking about the formation of diploid cells, sorry, formation of haploid cell from a diploid cell. So it is best way to understand this by oogenesis, that is formation of the egg cell. So we have this diploid oogonium, okay, which will undergo mitosis to form the primary oocyte, which is also diploid in nature. Okay, that is your primary oocyte. Okay, now these egg cells, before entering the meiosis, they are diploid. Fine. So now what happens is these cells, okay, they will enter the meiosis I. Okay, so in meiosis I, we will get two cells. That is, what is the first polar body and the secondary oocyte. This first polar body may divide further to give rise to haploid cells. Okay, further give rise to haploid cells. Okay, and to form the polar bodies which usually die. And the second one, that is the secondary oocyte, which is also haploid in nature. Okay, it will further undergo meiosis II. Okay, to form the ovum cell. Okay, which will get released during the ovulation, further giving rise to your mature cell. Okay, now this meiosis II, okay, the secondary oocyte, it will undergo meiosis II to form one ovum or the egg cell, and second one the second polar body. Okay, the ovum cell will get matured into an egg cell, which will further enter your fertilization. If there is fertilization, then it will form the zygote. Okay, so now here you have to remember one important thing is that during the prophase I, okay, your egg cell, okay, it will get arrested. Okay, and then when the puberty is reached, again it will start the process of ovulation where it will undergo the second part of your meiosis. Okay, this thing you will study when you will study about, or you must have already studied about the human reproduction or the fertilization process. Okay, now let's move on to the next phase, that is, we have seen what is prophase I. Okay, then we had taken the example of oogenesis. Now let's start with the leptotene. Okay, what happens in leptotene phase? Now the term lepto, okay, it means thread-like or something fine. So from this term lepto, we should know that we have to introduce the thread-like structures over here. So here the chromosomes, they become visible under the microscope as the thread-like structure. So here condensation of the chromosomes continues. So if you remember mitosis, prophase of mitosis, we know that there is condensation of chromosomes occurring. So same thing is happening over here. Okay, that the condensation of chromosome will continue. So next phase is your zygotine. So here you have to remember zygotine. So there is this zygote term here. So if you remember what is a zygote? Zygote is the union between the egg cell and the sperm cell, right? The product of fertilization of your egg cell and the sperm cell. So zygote. So here you have to remember there is something or the two things are coming together giving rise to this zygotine. Okay. Now here, so here your chromosome will start pairing together. Okay. This homologous to form homologous chromosomes. Chromosomes will start coming together. Okay. And this process of association or coming together of your chromosomes is called as the formation of synapses. Okay. So these paired chromosomes which will come together, they will form the homologous chromosomes. Okay. Now this synapses, what is formed, okay, it is associated with something called as formation of synaptonemal complex. Okay, so what is this synaptonemal complex? Synaptonemal complex is your, okay, a formation of a proteinaceous structure, okay, or a proteinaceous complex. All right. So this is something new. Two things we have introduced over here, rather three things, that is synapses, okay, there is synapses, okay, homologous chromosomes, and now the new thing that we have introduced over here is the synaptonemal complex. So these are the three things that we have introduced now in your zygotine phase. So if you see this picture, okay, they have a line lengthwise, there is pairing, okay, to form the homologous chromosomes. Now this homologous chromosomes, they will form something called as the synaptonemal complex, okay, Okay, so it is a proteinaceous complex formed between the two chromosomes. Okay, now let's see the next phase. So next phase is the patcheting. So what happens in patcheting? Here again we are going to introduce different more terms. Okay, more different terms. Now here we have a bivalent chromosome. Okay, it will appear as a tetrad. So what is a bivalent chromosome? So if you see here a bivalent chromosome or a tetrad. So when we say it's a tetrad, when there are four different things involved. So when I'm saying four different things, here there are four sister chromatids. Okay. Now if you see here, okay, these two arms of chromosomes, they will form the sister chromatids. And these are this one, okay, 1 and 2. These are going to be the non-sister chromatids. Okay, so the bivalent, okay, these are the homologous chromosomes. Okay, they have associated or come together with the help of synaptonemal complex. They are, okay, paired. Now this leads to formation of a bivalent, okay, which is, or it is also called as a tetrad because there are four different things involved over here. Okay, all right. So basically your, the synapse chromosomes which are formed, okay, these are going to be physically connected during meiosis. So that is called as your bivalent. Okay, so this other picture will see later. All right. So I have introduced what is a bivalent or a tetrad. Now at this stage, there are some very important things happening that, there is something called as a recombination nodule. So it is a site where there is going to be crossing over between the non-sister chromosomes. Now here we have the homologous chromosomes aligned. Now we have seen, I had already told you what are the non-sister chromatids. Okay. Now there is going to be, crossing over or the exchange between the two non-sister chromatids. If you see this part, okay, there is crossing over between two sister non-chromatids of the homologous chromosome. Now let us consider one conference. Okay, say a biology conference. So in a biology conference, there will be people, okay, from different aspects of biology, from different fields of biology, where they will come to that conference, they will exchange their thoughts. Suppose we have person 1 from say, uh, background of say zoology, okay, and if there is another person, okay, who is from microbiology, okay, which is also a part of which it falls under biology, basically life sciences. So these two people are going to exchange their thoughts. They have, though they are from the similar field of biology, they have different knowledge, okay, they have different subject expertise. So when they are going to talk to each other, when they are going to share the information, there is going to be an crossover of information. So crossing over, it leads to recombination, that is combining different information. So crossing over causes recombination of genetic material on two chromosomes. Same thing that happens in a conference, right? So crossing over is basically exchange of genetic material between two homologous chromosomes. Okay, all right. So this happens because of an enzyme called as recombinase, which causes this crossing over. So this recombination between homologous chromosomes, it will get completed by the end of this patch-tin. And now what we have is called as, or it can be called as recombinant chromosomes. These recombinant chromosomes are the product of this crossing over. Now this crossing over has occurred between two non-sister chromatids. So if you see here, there is crossing over. Okay, there is an exchange of information. All right. So coming back to this picture, we have this recombinants, which are having, there is an exchange of information. We can see from this color coding, we know that these two recombinants, whatever are formed, they have information from different sister chromatids. Okay. So let's move on to the next phase, that is diplotin. Okay. Now what happens in diplotin? In the previous case, we know that there was crossing over. Okay. Suppose now this is one sister chromatid. Okay, this is one sister chromatid and this is another sister chromatid. So there was a crossing over between the two sister chromatids. Okay. Now, the beginning of diplotin, okay, there is dissolution of the synaptonymal complex. So, the synaptonymal complex, what had happened over that had formed, okay, it gets dissolved. So, dissolution, and we can correlate it to disappearance. Though the spelling of disappear and diplotin is different, but it sounds like disappearance and diplotin. So we can say that there is dissolution or the disappearance of this synaptonymal complex. Okay. Now the bivalents, okay, the recombinant homologous of this bivalent, they will start separating from each other. They will be free, but except at the point of your crossing over. Okay, they will get separated completely, but except at the point of crossing over. So if you see this picture, they are still attached over here. Okay. So what happens is, okay, I think there's some problem with the slide. No, it's back. Okay. So the synaptonymal complex, it is going to dissolve, but except at the side where there was crossing over. So it is still attached. Okay. So now here we have something called as X-like formation. So this X-shaped structure is called as the chiasmata. Okay. It is called the chiasmata. So now in oocytes of some of the vertebrates, this diplotin can last for months or some years. So in case of diplotin, we have introduced some more terms, okay, that is chiasmata, okay. So in the previous case, we had introduced recombinase enzyme, recombination, okay, bivalence, homologous chromosomes, and now one more term, that is your chiasmata. So it's a X-like structure. Okay. So next phase is your dikinesis. So your dikinesis, it is the final stage of prophase I of your meiosis. Okay. So now in case of dikinesis, so we can say di, something is getting over. So what is getting over is your chiasmata. So there is terminalization or disappearance of this chiasmata. So now there is no chiasmata. Your chromosomes are free. Okay, recombinant chromosomes are free. There is no association and they are fully condensed now. Because if you correlate this with your mitosis, okay, what was the end product of your prophase I? So they were completely condensed chromosomes which need to get attached to your spindle fibers, because in your metaphase they need to come together or get aligned towards the equator of your metaphase plate. Okay. So they will start preparing for your separation now. So by the end of dikinesis, your nucleolus is going to disappear. So if you recall your mitosis, prophase, so again there also there was disappearance of your nuclear envelope, then your different organelles and nucleolus. Everything had disappeared. Same thing is happening in case of your prophase 1 But at the end, the last part of your prophase 1, that is your dikinesis. So from dikinesis, your cell is going to shift to metaphase. Okay. All right. So let's see what happens in case of your metaphase 1. So in your metaphase 1, okay, the bivalent chromosomes, they are still bivalent, okay. Your bivalent chromosomes, they will get aligned on the equatorial plate, okay. It's the same thing that had happened in your metaphase of your mitosis. Now microtubules, they will start forming from the opposite pole and they will get attached to the pair of homologous chromosomes. Then next phase is your anaphase 1. So what had happened in the anaphase of mitosis? That there were separation of chromosomes. But now here there is going to be separation of your homologous chromosomes because they are still paired. Right? Now the sister chromatids, they will remain associated with the centromeres. So same thing had happened in case of your anaphase of your anaphase of your mitosis. Okay, there was splitting or the pulling apart of your chromosomes. Now here in case of your meiosis, we are talking about the homologous chromosomes. Okay, and the last phase is your telophase. So what happens in your telophase is the same thing what would have happened or what had happened in your mitosis, telophase of your mitosis. So there was reappearance of your nuclear membrane and the nucleolus, which will be followed by cytokinesis. But the difference is, in case of your mitosis, we had the diploid cell. Two diploid cells formed, but here we have haploid cells. So we call it as dyad of cells. Okay. So what do you mean by a dyad of cells? Okay. So dyad is basically double chromosomes resulting from splitting off of a tetrad. We are talking about the bivalents, right? So a dyad is a double chromosomes resulting from splitting of your tetrad during the meiosis. Fine. So there is a phase between the two meiotic divisions, that is meiosis I and meiosis II. It is called as the interkinesis. Okay. So this interkinesis is a short phase. Now soon after your interkinesis, there will be prophase II. Okay, and this prophase II is much simpler as compared to that of your prophase I, because under prophase I we had so many subcategories of your leptotin, zygotin, pachytin, diplotin and dikinesis, whereas prophase II will be very simple. So what are the things that we have studied or we had seen in your meiosis I? Okay, let's try to quickly recall. Again, it is subdivided into four categories or four stages. That is your prophase I, metaphase I, anaphase I, telophase I, followed by cytokinesis. The major difference is that there will be, transition from diploid to haploid cells. Okay, then we had seen what happens in your prophase I. It is more longer and complex as compared to the prophase of your mitosis. It is made up of 5 more subdivisions, okay, or subphases that we had seen. Okay, what happens at each phase? We had introduced some more terms which were not there in case of your mitosis. Okay, so what are these terms? So we spoke about something called as homologous chromosomes, haploid, diploid, synapses, formation of synectonemal complex. Then there was something called as bivalent or the tetrad. Then we spoke about the crossing over or the recombination. We spoke about recombinase enzyme. Okay, then formation of chiasmata. Okay, these are the different new terms that we have introduced, and each term requires special attention, especially when you are preparing for your MCQs in NEET, because this could be confusing. So you have to form a logic while you are studying. So then we saw what happens at each phase and what is the transition from prophase I to metaphase I. So from metaphase I onwards to your telophase I, everything will be same, okay, as compared to the phases of your mitosis, but we have to speak about the homologous chromosomes. Okay, so here at the end of your telophase, there is cytokinesis giving rise to dyads of cells, which will have two cells with haploid makeup. Then there is the phase between the two meiosis, subsequent meiosis is your interkinesis. Okay, this is what we have seen in your Meiosis I. So in the next session, we will talk about Meiosis II. So till then, keep studying and keep smiling. Thank you.